Buonasera, benvenuti a GiovedìScienza.
Good evening, welcome. Welcome to GiovedìScienza, Science on a Thursday. This is a special one, and this is the fourth year that we present the person who won the GiovedìScienza Award, the prize. The popularization of science always presumes a good scientist. But popularization is something you have to start off with when you're very young. And it is part of the work of a scientist. These scientists, mostly, especially in the early stages of their lives, usually is funded by public money. In the US, this is considered a good reason for them to give back the results to the public. It's something that is starting in Italy. It's working quite well for the past few years. At the University of Padova, I in fact teach a course on scientific communication for doctoral students and postdoctoral students. 4 years ago, we had this idea, and it wasn't mine, it was my wife Eleanor's, to have a GiovedìScienza Science on a Thursday prize for youngsters who were good at research, but also who were interested in popularization. These are the two qualities that we seek in awarding the prize. The first thing is that we shortlist them on the basis of their scientific publications. And over the past 4 years, we have been able to appreciate the high standard of the Italian young scientists. And then there's a moment when you also have to assess their ability to communicate. This is something that we do by selecting amongst the various entries, who are about 50, 60, the best, as I was saying, are shortlisted on the basis of the publications. Then the top 10, then asked to hold a presentation in front of 100 students and a jury. And they have the same weight as the youngsters' jury, and they're basically all very good in terms of their ability to carry out research projects, but we see which one of the 10 is the best popularizer, and on that basis the person wins the GiovedìScienza prize. And we hope that this has a big future, especially for the whole of Italy. Today we have Alberto Massarotti with us. He works in Novara, and he works in pharmaceutical or drug technology at the University of Eastern Piedmont, the Avogadro University. He works more on the computerized side. He works on molecules to identify the ones which possibly might to be developed into drugs as and when they were processed by other researchers. It's a very useful job. It's also a sort of discipline, a new scientific discipline, very recent, very important, because don't forget that out of about 100,000 molecules, which may have some results in terms of production of drugs, not more than one actually reaches the chemist or pharmacy. And there are many mistakes and many problems, and this is clearly a very expensive process. Now, big drug companies, the big pharma, have their faults, but certainly they do have a lot of waste in this. Let us take the situation of antibiotics. Resistance to antibiotics is growing and has been growing in the past decades, so it is necessary to find antibiotics to replace them. And in recent times, it has been very difficult to find them. It's a very important activity in terms of the improvement of good health and also of saving money, avoiding to undertake research in areas which are dead ends. So I'd like to give the floor to him now. He won the last edition, the latest edition, in fact. Now there is a call for applicants for the next edition, but before he tells us about the work he's doing, I'd like to start with two questions. I'd like him to tell us how he got to the prize, because one of the things is also we know that it's quite difficult to become a researcher in Italy. So the floor to you. And first of all, congratulations. Thank you very much.
Io ho iniziato studiando come biotecnologo, mi sono laureato a Novara.
I began by studying biotechnologies. I graduated in Novale in medical and pharmaceutical biotechnologies, following which I decided to start a PhD in computational chemistry in the group of drug or pharmaceutical technologies. I did this because I love IT, and the professor thought that I might be thus inclined to discover new molecules, which is what I will be talking about. Let me say that I know the professor. It's Professor Giovanni Appendino, who is extraordinary, especially because he favors the development of young people, the ones who surround them. Oh, yes, he was my tutor, and then I went to work Professor Sora and Professor Trona's groups. In the course of my doctoral studies, I was not only in Novara, I also went to Cardiff, to the School of Pharmacy, and that's where I discovered the haptic, which is the instrument. It looks like a coffee pot. Yes, it looks like a teapot or a coffee pot, and you will be explaining what it is all about. Yes, and I will show how it works. And at the end, you can try. It's called haptic. It's haptic because it comes from the Greek one, that is the tactile feedback. In fact, it might seem a coffee pot or a pen, but this is a discovery in Cardiff. Yes, they started using it in Cardiff to use it with proteins and molecules. Haptic was originally developed for video games, and then this model has a pen-like handle because it's used for 3D designing. So it shifted from the game area to research. Yes, this is also curious, which has also meant that it's much cheaper because since it was for games and largely sold, the cost dropped dramatically. And so this is an advantage for everyone, for those who work on it and for those who play on it. So you came back to Italy, and when I came back in Italy, I finished my PhD. I stayed in the same group for a postdoc for 3 years, following which I became a fixed-term researcher at the Department of Drug Development in Novara. The next question is it the first time that you had taken part? No, it was the third time. You must be under 35, so you started very young. You have about publication? No, about 30. Well, 30 publications, one a year, that's roughly. I think that's quite good. So this is Alberto Massarotti. We've introduced him. And now let us see what he has to explain. I'll be talking to you about how drugs can be designed on the computer. This is the department where I live, but where I work. But before doing this, I would like to define some of the words I'll be using, in particular drug design. Yesterday I was interviewed in view of today's conference, and the radio conducer said, how many drugs that I had designed were sold in a pharmacy. Well, if you thought that you were coming here to find the answer, you will be disappointed, because you will find any of the drugs that I have designed in a pharmacy. So there's a problem of what I mean by design and what might be the common use of the word. For example, let us think one of the most common drugs. I think about paracetamol. Paracetamol, it's the one that we have at home, but if we see the definition of it, it's any organic or inorganic substance able to generate functional, useful, or damaging changes through a chemical, physical activity. This is the definition of drugs. And I think that the difference between what I think and you think is enshrined in the word design, because we consider it as an engineering, very often, definition in car design. Each part which you design has been conceived and manufactured knowing exactly what was the final object, that is to say, a car. If we want to improve on any one of those components, we can do so because we have the exact idea of what a car should be. The problem of designing a drug is that in our case, we are thinking about designing it for a living organism, human beings, but not only, but a living being. However much we study it and however much we know it, is really a very complex machine. So we are unable to modify or improve a molecule having an exact idea of all the effects that it might have on the organism. In fact, drug design is a process which involves a number of disciplines. Here you can see some. I've listed, we need organic chemists who can design new reactions, pharmaceutical or drug chemists that can identify active principles, analytical chemists to analyze them, pharmaceutical technologists, biochemists, biologists, toxicologists, right up, pharmacologists, and then doctors who will then be the ones who decide whether an active principle will be useful to solve a problem or not. About 14 years are the time required if you go from the first molecule, when you've identified them, starting with 100,000 molecules, to identify the one molecule which will be then marketed. So when I talk about drug design, we're thinking about what we're doing at the beginning of that process, in the early stages. That is to say, how can we select or improve our ability to select those 100,000 initial ones. So my work is in the initial stages of this process to develop new molecules, which we hope will in the future become new drugs. But we're like athletes, like trainers. We're trying to identify which molecules are the ones which show features that suggest that they may become drugs in the future, but we're not the ones that will take them to the race. We're just the ones who select them. So this is a metaphor. So it's like selecting the junior team of a football or soccer team. Yes, a bit like that. Yes, in a certain sense, that's what it is. What I do in practice, since I'm in the computational field or the IT, I choose the ones which are most promising, that I then pass on to those who are involved in synthesis. Computational drug chemistry includes a number of different techniques and possibilities that can be used to select compounds. Today I will be illustrating some which I think might be most interesting for you. My field of work became more important, especially after 2013, when Karpus, Levitt, and Warshall were awarded the Nobel Prize precisely for computational chemistry. The motivation was for the development of multi-scale models for complex chemical systems. In very simple words, what they did was to use the principles of physics to simulate the movement of the atoms in molecules, and therefore were able to simulate a reaction catalyzed by a protein. This was 2013, but the work that they was given for was of the 70s. And here I'm showing you the first page of an article which they published in '75, and an advertisement of the type of computers that were being sold. At the time, there were no floppy disks. They were about to create Apple. Those were the years. And the supercomputers of the time, this was marketed in '76, were of this type. Probably looked more of the waiting room, that is to say, where you think you could sit down was the cooling system for the processors for the calculations. It's also interesting to notice that a modern mobile is 10 times more powerful than that supercomputer. This gives you an idea of how much the technology has progressed in terms of speed, and this is also one of the reasons why in my field we have been able to use it and apply this power to drug design. But here you see the computational chemistry in its early days. The Nobel Prize is especially for the incredible efforts that they had in those days to apply the instruments of those days, available then, to what we're now also doing. What is ITC or IC useful for? Well, first of all, virtualizing reality, because the biological context is much smaller than what we can do, and then to repeat things, which means that we can handle a lot of molecules repeatedly. This is the photograph of the box of the drug, but in fact a drug is a chemical structure. This is also virtualization. With a computer, what we can do is that we can virtualize the structure in a different way. It's the same chemical structure, but we start to give it more depth, because otherwise you don't have an idea of what the size and dimension of a drug are. The third one, you can see that each atom is a sphere, which depends, the size of which depends on the radius of the atom. This is the atom. This is paracetamol. So we have to think in 3D. To give you an idea of how they will interact, I chose paracetamol because I think that paracetamol is a very common and well-known. I went to see an example of how paracetamol can interact with the protein. Here you see the structure is 3D, because this is the position that paracetamol takes within a protein. In blue, you can see the amino acids that are the protein, which is the target for paracetamol. In purple, you see the M group, which is a molecule that is inside the protein, and paracetamol, in this case, interacts with the heme group. Isn't it the one which is the center of hemoglobin? Yes, it is. Yes, it is an iron molecule at the center. It's a heme. This is a virtualization, and you will observe there is a lot of empty space. In fact, it's an image which enables us to see the center of a protein. We could also represent it with the surface of the protein, and immediately you will realize why we call it a cavity or a pocket, because this is where molecules can enter. It's a cavity which is a very small part of the protein as a whole. Now, this structure has been virtualized, but it is not computer generated, it's computer visualized, and it's based on actual data. What do we need to virtualize reality? Well, we need biochemists to crystallize proteins, which will then shift the X-rays and show us the protein structure, which is what was done and which made it possible for us to have this structure, which is a file and which is a representation of reality in scale. These are publicly available. There's a public database, the Protein Data Bank. To give you an idea of virtualization, I printed with a 3D printer a protein. Yes, we have a 3D printer. This is the model. To give you an idea of the size, this is a model which is 10 million times bigger. It's a scale of 1 to 10 million. The size, here this one is 8 centimeters, but in fact it corresponds to 80 Armstrong, and that's 10 to the minus 10. Yeah. So in the image, I don't know if you can see it, the binding site is completely internal. I also cut the model just to give you an idea of the position of the molecule. It is a sort of fitting in one in the other. It's not only like Lego, but the model which we use to explain, it's called a key and lock model. It's a very approximate idea, but it conveys the idea that the molecule, which is the key, must interact within the protein, which is the lock. So that red one which we saw there, that could be the lock. Yes, in this case it's a heme group. Its paracetamol is even smaller because in proportion, that's the group of molecules which paracetamol goes into. Yes. You see, here it is. It's like a Lego. And this is the iron, which is very important for oxygen and for breathing and to burn food. Keeps us alive, basically. Yes, it's the oxidation between the two states of the iron which keeps the mechanism ticking. It was something that we didn't know about before. Yes, this model is not. This can explain the proteins, because proteins, one must bear in mind that they are not static, they keep moving. But it gives us an idea of how the biological mechanism might work and how it might develop an action. There must be an interaction which clearly depends on its form and how molecule and protein can interact. I work with a computer, and one might think that to discover new drugs, we just need all the new keys. Let's try and work it out. Let us take 5 different atoms: carbon, hydrogen, oxygen, nitrogen, and phosphorus. We try to combine them in all the possible ways and select only the molecules that are below 500 Daltons. Take 500 as a threshold, and I will not explain to you just now why we chose this threshold. We might obtain 10 to the power of 60 molecules, which is an incredibly high number of molecules. To try and understand it, because we're not able to conceive numbers as big as this, I wrote it out. It is 1 with 60 zeros. We call it the chemical space, to give an idea of how big the number of molecules is, the number of molecules that we might generate. Clearly, this number includes a lot of molecules that might be very good drugs. To give you an idea, the difference between chemical space and real space, space as we know it has existed for 14 billion years. Okay. Let us imagine that there is a chemist that for each second since the universe was discovered has synthesized a molecule. Well, we're not even at 1,000th of 10 to the power of 60, possibly only 10 to the power of 20, which is very distant because we're talking about a factor of 10 each time. Yes. So there are people who have estimated that it says that if we wish to synthesize all molecules, there wouldn't be enough matter in the universe. Because all the particles of the universe are 10 to the power of 82, 81. That's including all the particles, including quarks and electrons. So imagine 10 to the power of 60 is a figure which tends to be quite closer to the total. And it's a number that, however powerful the computer is, I don't think we'll ever be able to handle. Here what you can see is an outline of our known chemical space. In this case, it is a bidimensional presentation. Here you see of a 3D space, and this we also have a space of right up to 42 dimensions, which gives you an idea of how complex it can be when we're dealing with the molecules that already exist. However, we have to find a way to explore this chemical space, which is something that we can do by virtually screening it. How do you do that? Well, in this case, you screen the known chemical space. This is done using different filters. Each filter that we use reduces the number of molecules which are allowed to move on to the next stage, so that we then get to a number of molecules that can be handled and that may in the future develop as drugs. So this is an example. I worked on tubulin. Tubulin consists of 2 subunits, and again I've also printed the models to show you how they work. Tubulin is something which assembles, and you can see it, that it can be done like this, and forms 2 subunits. It's again like Lego, because proteins have a shape and they're complementary to each other, they complement each other. And this is known as alpha and beta tubulin. Why is tubulin important? Well, tubulin is a dimer which polymerizes and forms a microfilament, and the microfilament in turn can be assembled in more complex structures which are microtubules, and this sort of a tube. And there are 3 binding sites where there are molecules that interact and that are able to block the formation of the microtubule. Blocking it, or in any case, the microtubule can't work. Why is it important? This is a representation. It's not the photograph of a microscope, but it gives you an idea of the section of a cell. And if you magnify it, you see that inside there are proteins. We could talk about it in terms of it being an ordered chaos. A new microtubule is the one which structures and gives shape to other proteins, and it is particularly important in mitosis for the mitotic spindle. It is the one which stops the reproduction of cells, and this is very important in cancer and tumor because they're hyperactive in cancer and they duplicate more than normal cells. If we're able to block it, then we can produce molecules that will have an anti-tumor effect. So what we decided to do is to find molecules that were able to interact on colchicine. This was downloaded from the protein database. This is a drug used for gout, and the colchicine gives the name to the site, which is at the interface between the 2 subunits. Has this got the name of it to do with the plant? Is it a plant origin from Colchicum autumnalis? Yes, it's a plant that we all know. Yes, it's a natural product. Certain seasons, after winter, or in the autumn, is it? Autumn, yes. This molecule, as you can see here, this is made simplified for you to understand it, and since it's at the interface between the two units, it disturbs the shape that the alpha and beta tubulin may take, and the way in which they bind impacts on the microtubule. We went to look for this in literature, and we found other molecules. Here you have a large number, 15 represented, that are active on the same site. What we did, we also saw what were the parts of these molecules that were shared, and we identified the ones which are called pharmacophoric groups. That is to say, they're functional groups and parts of chemical structure which are common to all these structures. We identified 7. To give you an idea, this is again colchicine that we saw before, and the sphere represent the positions that these groups, which I've just mentioned, may take. What is the aim of doing this? If these 15 molecules have these common pharmacophoric sites, it is very likely that other molecules that have the same sites, we don't know whether they'll be active or not, will also have the same effect on the structure. We worked with a computer, which means that we can measure from the protein structure the distance between these various pharmacophoric groups, and we transform this in a model that it can be processed by the computer. We then use this pharmacophoric model as a filter for virtual screening. What does this mean? That in this phase, you write programs or you use program. In this case, we used a program that already exists. In some cases, we have to build algorithms, which are the simpler forms of the programs that make it possible to optimize what we do. In this case, we had an available program, and the only thing we had to do was to input the data, to select what we were interested in, and have a model generated. At this point, we had to select the chemical space, the known chemical space that we wanted to explore, and we identify a database of existing and marketed molecules, about 500,000 compounds. The aim that we had was to test the molecules to see which ones could be further developed. When you process this information, the first thing to be done is to filter them with a one-dimensional filter. That is to say, that it doesn't go to see the three-dimensional information like the one that we've seen, but it just measures parameters. Parameters which normally are Lipinski's rules. Lipinski used to work at Pfizer's, and he noticed that the molecules that became drugs and that were developed to absorption or uptake by mouth had the following characteristics. So molecular weight below 500. They had a lipophiliac below 5.0. They had a certain number of hydrogen donor bonds below 5 and acceptor. Well, this is the sort of identikit, isn't it? Yes, we say that 90% of the drugs fell under these parameters. So reasonably, if we develop these parameters to select molecules, we have a 90% possibility of identifying molecules that may become drugs in the future. Because if they're too big, the parameters, in particular pharmacokinetics and absorption, that are optimal, and that means that they can be absorbed, because when we absorb them, they must also be able to reach the target. Tubulin is within the cell, so it means that the drug must be able to penetrate the membrane, which means that they have to have the properties which are necessary to get to the target, to reach the target. So the first thing that we do is to identify the molecules that answer these properties. As I mentioned, molecules are not static, and they may have more than one shape or conformation. They're complex structures, and in particular, they have bonds that can rotate. The position or the shape that they attain depends on their organization in space. So we carried out a conformational analysis where we generated all the possible stable conformations that molecules might have. Here I have then put the 40 structure, the 40 most stable structures of the example that we gave, and you see that they occupy different positions in space. This is useful to us because when we then filter them with our pharmacophoric model, we are sure that we're actually working with the conformation that the molecule can take on. So in the end, the molecules were analyzed, selected molecules were analyzed, and 44 were identified. These compounds were purchased, they were tested, and 2 showed that they had an activity around about 200 nanomolar. At this point, molecules were passed on, so to speak, to the people who do synthesis, and we managed to optimize it and find compounds, in particular 2 that were particularly active at 30. How do you translate these measurements? Well, the smaller the concentration that we can use for a drug to have a biological effect, the less the amount that we have to have as in the drug to have a biological impact. This means also maybe the side effects would be less. The smaller the quantity, the less the side effects. Well, that depends on the modes of administration and what the other targets are. In this case, we saw how it interacts with tubulin. We did not see, because it's not yet done at this step, it's done later on, what the negative side effects are. What we're interested in, we have to find a molecule, a lead, that has an activity on our target. Then, according to the type of modifications that we have, we managed to improve both the selectiveness and the power with relation to other targets. The information that we obtain is useful in this case both to develop further molecules and different classes which will target tubulin. Now, we had a known chemical space, that is to say, molecules that already existed, and which to a degree meant that we gave a second life to this molecule. Now, it might also be useful to analyze new areas of the chemical space that are not known, and this can be done using virtual databases that do not represent the entire chemical space, but represent slices or parts of the chemical space that are accessible, because not all the compounds are known, and not all meaning that they can't known how to synthesize them, because it's ridiculous to have information on something that we cannot obtain. So in this sense, we decided to create a virtual database of molecules that were synthetically accessible. In 2001, another Nobel Prize for Chemistry was given by Professor Shardless on the chiral catalyzers in oxidation. In particular, here you see him. He introduced the notion of click chemistry. You can see the example which he has. This is the most famous click chemistry or click reaction. It's a reaction that gives a high yield and which generates a product which can be easily modifiable with a click. Plug and play, if we want to use an IT expression, and it is the cycloaddition of catalyzed copper between alkynes and azides. This is an alkyne and this is an azide, and we see that in the presence of copper we have a cyclic structure that's known as triazole. The reaction, I've got photos of it here, can happen in a vial where you see there is a copper wire, and you can also shake it because in time there's a white powder which is the final produce and which can be filtered to obtain the desired compound. Triazoles are not simply a connection between other structures, but it can interact with proteins, which means that hydrogen bonds and hydrophobic bonds can be formed. It has a role with our target, with our protein. What we did is that we generated a galaxy of triazoles, a galaxy which we know is linked to the click reactions. Here we downloaded all the ones that had already been downloaded and created. In this case, we wrote the program to have a virtual interaction in the computer and generate 16 million triazoles. We have a library of compounds. It's all virtual, which covers spaces of the chemical space or parts of the chemical space which do not overlap entirely with the existing molecules. And in this representation, we have orange, which is the part of the chemical space covered by our database. And then you see the database in blue of the ones which are already marketed, and yellow, the ones which are already drugs. Our database includes a vaster chemical space as compared to the one which is occupied by drugs, but it overlaps partly. So we have also basis or to hope that we will be able to develop molecules that are potentially new drugs. We then went to see how many molecules existed in literature. Of our 16 million compounds, and only 320 compounds have already been synthesized and tested on biological targets. So we have a lot of leeway to find new compounds that haven't been patented and that are therefore not covered by copyright laws or intellectual property laws. So here we have this database. Anyone in the world, any researcher who wishes to use this database can do so. They can download it using them in their products and in their projects. We've shown two ways to explore chemical space, and what if instead of exploring it, we decided we'd like to make new molecules, de novo design? Well, what I explained in my presentation at GiovedìScienza is the application of the multi-component reactions. Multi-component reactions are a type of reaction which requires 3 or more reagents. Here you see there are 4 reagents which react in a combined manner to produce one single compound, and that is chemically very diverse. What I developed is a way to get the computer to process this information so that it was able to learn as a brain, to understand and the chemical structures, to develop them, and to design new molecules that would benefit from the interaction. Now, human brain memory and learning is based on neurons. Neurons are stimulated by outside stimuli, and that in turn generates a response or reaction. So we had to develop this to memorize information, enabling us to think and learn from stimuli. What I designed is a neuronal network, that is to say, a program which mimes neuronal behavior. In this case, my network, which is a program, was put in touch with all the chemical structures of the compounds which were already existing. And the program learned how to understand how these structures can be made or built. As a result, after this learning phase, the program is able, if put in with a new structure, to disconnect it or disassemble it in its components and tell us which reagents we should use to generate compounds. What we obtain is a code, a program, which starts with a molecule and is able to suggest a way of synthesizing it. The program can also be used in the opposite sense, that is to say that starting with the possible reagents, it can suggest how they can be combined to generate new molecules. These new molecules might be built in the binding site of our protein, because if the final aim is drug design, but the drug, as we have seen, cannot be manufactured just using the entire information of the living body, but we can use the information linked to the target, the biological target, that is to say, our proteins. How can we do this? Here you can see the representation of a protein with a green molecule inside the binding site, and what we can do is to simulate how the molecule will position itself in this pocket. It's called docking, molecular docking, and dock is like the one which you use with boats. Here you can see is the image of what can be visualized in the programs, and all the molecules that are tested in the building site. We simulate the positions that these various molecules may take. If we slow it down and we see it in detail, what is the program doing? It's using the formulas of physics, that is to say, with classical physics, so we can simulate and estimate the binding energy of the molecule within the binding site. So we have a number as a result. This is done for each molecule, and in this way we are able to obtain a value which will give us a priority listing and an estimate of the interacting between molecules, so that we can identify which ones are the best ones, because they have the highest probability of having a biological impact. A computer is able to simulate this on its own, but what we would like to do is, our idea was to also implement not only the formula of physics, but also the knowledge of a chemist working on this, able to guide this and to help choose the position of the molecule. To do this, we decided to use an haptic interface, the best known is a video game one. Haptic, as I said, because it is a tactile feedback. In the case of the video game, it's a vibration. If, for example, you're driving a car, but in our case, we're using the coffee pot haptic, which is very sophisticated, where there is a mobile arm which is used to move a molecule within a binding site. Now, I would like to show you how it works. Here you see we try to load now a protein, and we see how it can be moved within the site. This is the first type of representation. The red sphere represents what the molecule might become. And this is the representation of the protein. If you zoom in, our sphere, which represents here the molecule, when we get closer to the structure, the haptic mechanical arm gives me a tactile feedback. That is to say, I'm able to perceive the shape of the protein. Would you like to try? I don't know where to go to. you should soon start to perceive. You see, you can't move it there. Yes, that's true. So this is how you can use the experience of the chemist running this in directing the interaction. What we want to do is clearly not just move the molecule around that represents our ligand, but to see that our program that knows how to generate molecules should plan or design the new structures when we go to position it in the binding site. Thus, we're able to maximize the use of the program, but also the knowledge of the person who is running it and who is using it. What I felt as a resistance, what is it? Was it the electrical field between the molecule and the site of the protein? Yes, it was reduced because if you had to move a negative molecule against the negative amino acid, it would not be possible because it would throw you back, but you are able to feel it in a sort of subdued, dumbed-down way. Yes, the program does this. The software, I don't know if you notice it, but is able to tell you which is the amino acid which you're interacting with, and also see whether there are hydrogen bonds or other types of interaction that form. This might be useful, at least this is our idea, to design new molecules which shall have to be synthesized by a chemist to obtain new drugs. And, well, these molecules that we hope will become new drugs. To use the haptics or other technologies that come from the world of games has a name, and it's called gamification. In the last part of my presentation, I would like to show you two examples of gamification which are quite well known. These are not projects that we ourselves developed in Novara, but they're projects that we could all, if we wish to, join up with. The first one, which I'm going to show you, is Foldit. I showed you programs for proteins. Proteins have a shape, which means they're in space, but it's a sequence of amino acids. Amino acids and their sequences are positioned in space as if they were a filament or strand. Here you can see it much better than this other one. And here you can see you haven't got the surface, you're lacking it, and the protein must reorganize it to have a 3D organization, which enables it to have a biological function, which is the folding of the protein, which is how it folds, yes. The structures that have been deposited in the crystallized database are not all the possible proteins. This means that if we wish to see a protein which hasn't been crystallized, we must find the way in which we can start from the information which is inherent in the amino acid, because the final structure depends on the amino acid which makes it up, but which we cannot easily simulate or predict, nor are we able to predict how it will be organized in space. Foldit is the simulation of how the amino acids of a protein might organize in space, but it's also a game. This means that each and every one of us can install it and contribute to the hypothesis of how a sequence of amino acids will organize in space. Proteins are segmented, and each person playing will fold the filament and will have points according to the energy that this shape or organization may develop. If we have all the players in the world, we can then have a structure which was then confirmed by X-rays. So gamers are able, precisely because in a certain sense the organization of a protein has a degree of harmony, they're able to do it. This tells us what the energy is, and the player can perceive and reach a harmonic organization faster than a computer. And what you play with it? With a computer, you can install it with a mouse. You move the structure around, just like a game. And this fact that you spread research out among citizens, well, it means that everybody can participate in research. In the end, it's not really necessary to understand the ins and outs of it, is it? For the player, it might just be to have a high score. So it's the gamification of science. It's a new way of producing science. Some citizens can contribute just by playing. Or they can also be aware of the fact that they have given a good contribution. This has been used, for example, in astronomy too, to understand the signals, possibly even artificial signals that came from space. It was also used in the identification of galaxies, which looked like little dots, but in fact were galaxies, and were identified as such. It was very difficult to identify them looking at them one at a time. And by distributing these programs, they have been able to involve millions and, well, thousands and tens of thousands of people in this, and who spent time on the computer and did it. But you said you can also do it with a mobile. Yes, it's another project. It's called Folding at Home. And again, the aim is to predict the structure of proteins. But in this case, the contribution that we can give is our computer's computing ability, or our phone's computing ability. When we're not using it, it can work to contribute to understanding how a protein organizes in space. In this case, the computing power is used at night when you are working, when you're recharging, because otherwise you'll be leaving the battery flat out. And what is done is that your mobile, as your computer, can be fed in to this bigger computer. Yes, very similar to the one which was used to identify extraterrestrial signals. And yes, so it reminds us about how powerful our mobiles are and the computing ability, which is 10 times as strong as the old crate computers that we mentioned before. We must realize what we have in our pockets, because now things seem very normal to us. Technology has become something we are akin to and we're familiar with, but there's a lot of research and knowledge in it concentrated in a very small space. And another example of technology that can be used for this project is a PlayStation. The PlayStation 3 already had this program in it, so if you wanted to, you could leave it connected and give the calculating power to generate and simulate possible reorganizations of the protein. Are there results that we can give and to show that this was effective? Well, in the case of Foldit, they have been able to correctly predict the structure of some of the HIV proteins, which had been solved, but it wasn't adequate to be used, and so we understood the AIDS virus through, yes, of some of the proteins of HIV, and that is to say AIDS, which was subsequently crystallized, confirming that they had been correct. And the players did so in 2 weeks. Very good result, very good result. I have a last slide here, with which I would like to conclude. All we've seen is the generation of models, a virtualization of reality. This is, he says, here you see that Ben says a model must be wrong in some respects, else it would be the thing itself. The trick is to see where it is right. And that is the slide which I would like to conclude with. It's important to identify the right aspects of our model. That's the trick. And this is very important in terms of epistemology, too, because it means that you learn from mistakes, and in every mistake there is something good, and just like in all the good things, there is a mistake, and that you move in between these two opposite sides, between the mistake and the correct solution. So with this, you have today become acquainted with Alberto Massarotti, a young researcher who won the 4th edition of GiovedìScienza. You penetrated in his labs, and you've seen what he did, what he does, and you've seen the ease and the humility with which he introduced us to a very interesting and sophisticated type of research. Thank you very much.
Grazie. È stato bravissimo.